Amazing. Thank you so much, Leila, for this introduction. And we're going to start right away with Jennifer. Please tell us about Bettermilk.
Hi, everyone. Thank you, Ana, for presenting me. I'm super excited to be presenting Bettermilk today at the Bioinnovation Conference. So I will begin. Milk is the original superfood. It was our first meal when we were born, and now it is part of our daily cravings. Globally, the dairy market was valued at $673 billion in 2019 and is expected to exceed the $1 trillion mark by 2024. However, the way we currently produce milk is not sustainable. Dairy market is responsible for 1/3 of the planet's ice-free land, 10% of the world's greenhouse gas emissions, and for 1/3 of the water footprint of human activity. The dairy industry is also responsible for tremendous amounts of suffering. Therefore, we need to find better ways to make milk more sustainably and ethically. I'm Jennifer Côté, and I'm the CEO of Bettermilk, where we make milk with mammary cells for sustainable and ethical dairy products with no compromise on taste or texture. And now you're probably asking, how does she do it? Well, let me explain it to you. The power of our technology is the mammary cell. Here we can see a human mammary gland, which is pretty similar to what the cow mammary gland looks like. Here are the grape-like structures, which are called lobules, that host alveoli. And in these alveoli, there are mammary cells, which are more specifically called luminal alveolar cell. These cells are the mastermind behind lactation. They secrete milk inside of these cells, outside of the alveolus, and into the nipple to feed the newborn. Of course, there are many more cells involved in lactation, but I won't bore you with the nitty-gritty details today. So these cells are harvested from the cow, small quantities, without hurting the cow, and then are brought back to our lab and are immortalized, which means that they'll be living on forever. And it also means that we won't be needing to get cells from the cow ever again. Then, these immortalized cells are genetically engineered to produce more milk, and then proliferated in trillions of numbers and seeded into this bioreactor, which has the same architecture as the mammary gland of the cow, so that they have the right structure to produce milk. Here is our mammary duct that we have created. The cells are coating the bioreactor and are secreting milk inside of this hollow lumen without ever coming into contact with the food that is given. Here is in green are the clusters of proteins that we have produced from our cells, but don't worry, the milk isn't actually green, it's only for visual purposes. Another special component about our technology is that we can create custom dairy products, which means that we can instruct our cells to produce lactose-free milk, low-fat milk, or even milk with added proteins. This is more time-efficient and cost-efficient than if we were to remove the lactose or remove the fat or add proteins to milk produced on dairy farms. Speaking about costs, if we want to offer you these custom dairy products at a competitive price, we need to tackle the medium challenge. In cellular agriculture, 95% of the marginal cost is due to the medium. The medium is the food that we give to the cells to make them healthy and happy. Right now, 1 liter of this medium costs $7,500, and 1 liter of medium is approximately what we need to make 1 liter of milk. So can you imagine paying $7,500 for a pint of milk? I don't think so. Therefore, Bettermilk has tackled this challenge and has successfully been able to produce 1 liter of medium for 15 cents. And that will allow us to bring you the custom dairy products that you need, that you love, for a competitive price. This is the team behind all of this. This is me, I'm the CEO of the company. And this is Lucas, my co-founder, who is also CTO and responsible of genetic engineering. This is Nushin, who is our lead scientist at the University of Toronto and is responsible for the bioreactors that I've talked to you about. We have also academic partners, Alaka and Benoit, some mentors in the United States to help us with the business, and a team of marketing researchers that are tackling consumer attitudes research. I am Jennifer, and I'm really happy to present you Bettermilk, a company that makes milk with mammary cells with no compromise on taste, feel, or texture. Thank you very much.
Thank you so much, Jennifer. And Adam, we're with you next.
Okay. Good afternoon, everyone. My name is Dr. Adam Westbrook, and I'm the metabolic engineering lead at Genecis Bioindustries. We upscale organic waste into affordable premium chemicals using proprietary bacterial cultures. So we probably all have a love-hate relationship with plastic. It's durable, it's very versatile, it's cheap. However, it's basically forever. And the promises made by plastic producers about being able to recycle them are largely been unmet. A great example of the impact that plastics have had on our planet is the Great Pacific Garbage Patch. So this is literally a small continent of plastic between Hawaii and California. It's now over 1.6 million square kilometers, so that's over twice the size of Texas. It's literally a continent. And some other fun facts, just in the US alone, in 2018, there were 36 million tons of plastic produced. Less than 10% of that was recycled. And 27 million tons ended up in landfills, which is almost 20% of solid municipal waste. Now, in response to the plastic glut, corporations have been urgently trying to replace petroleum-based plastics with bioplastics. And the great news is that we have had these around for much longer than the human race even. I'm specifically talking about polyhydroxyalkanoates, or PHAs. And these are often considered the platinum of the plastic industry because they're biodegradable. We can tweak the composition so we can use them in any number of applications, and they often offer better physical and chemical properties than petroleum-based plastics. But of course, there's a problem, or there has been historically, which is the high cost. And that is what we are trying to address at Genecis, and have been doing so since our inception. So traditionally, PHAs are made from oils and sugar feedstocks derived from sugarcane, canola oil, and corn. Obviously, this is a problem of sustainability or lack of sustainability, and also poor economics, because we're competing directly with food crops, and we also would need enormous arable lands to keep up with our insatiable plastic demand. So not really practical in the long run. Now, at Genecis, we have been trying to address the issues that are hindering the adoption of PHAs as plastic substitutes. We've developed novel technology to produce PHA for mixed organic waste using mixed microbial cultures, and we anticipate about a 40% reduction in production costs compared to other processes using the more rich feedstocks like corn and sugar. Now, our patented process actually comprises 2 fermentation stages that rely on trade secret bacterial cultures. We've been working on for about 3 years. So the first of all, volatile fatty acids, or VFAs, serve as the carbon source to produce PHAs. Now we produce VFAs in stage 1 of our process through anaerobic fermentation of organic food waste. Once we produce those VFAs, we can then filter them and feed them to another type of mixed microbial culture, which will convert them to PHAs intracellularly. And then we can harvest those intracellular PHAs to be used in end products produced by various companies. Now, we're currently focused on a specific copolymer called poly-3-hydroxybutyrate co-3-hydroxyvalerate, or PHBV. And the monomers are 3-hydroxybutyrate, or HB, and 3-hydroxyvalerate, or HV. Now, the composition of our PHBV is tunable. What that means is that we can change the HV content by modifying our fermentation parameters. And this will allow us to tailor the content, and then we can use the PHBV in any number of applications depending on the fraction of HV in our polymer. For example, for low HV content polymers of around 5 mol%, we can produce 3D printing filaments, mechanical pencils, golf tees is just a few examples. And then if we increase that to say 20 or more mol% HV, then we can use, we get much greater flexibility, which means that we can make thin films and things like that. Now, in parallel with PHBV production and mixed microbial cultures, I'm also engineering bacterial strains to produce PHBV with the goal of having tighter control over polymer properties and yield. And of course, in addition to that, we're also engineering strains to produce PHAs that are of higher quality, or sorry, higher value, that are sought after in the biomedical field, such as poly-4-hydroxybutyrate. And we're also going to extend this approach down the road to engineer bacteria to produce all kinds of value-added chemicals like antibiotics, amino acids, vitamins, other specialty chemicals, again, from organic waste materials. And finally, our technology has been verified by companies such as BASF. In 2018, we won an innovation challenge with them. We just won a big challenge, 2020 Novo Nordisk Innovation Challenge. We came in first place. On top of that, we're the youngest company to receive a $2.3 million grant from Sustainable Development Technology Canada, or SDTC, and we won other numerous awards as well. And that is all for me today. Thank you.
Thank you so much, Adam. That's really a good example, a great example actually, of the circular economy. And before we start with questions, we'd like to hear from Nivatha as well. So pass it over to you, Nivatha.
Hi, everyone. My name is Nivatha. I'm the CEO and founder of Dispersa. At Dispersa, we focus on the surfactant industry. You might wonder, what are surfactants? These are a type of specialty ingredient that are actually found at the heart of many soapy products that we use each day, especially during the pandemic, such as cleaning products, personal care, cosmetics, oil and gas industry, agriculture, and more. The surfactant industry is actually valued at nearly $60 billion, and this is due to its vitality in a lot of products and processes. Annually, we see that the global surfactant production is estimated to be over 20 million tons. Now, as we venture into the surfactant market and really break this down, we see three main categories: that are petrochemical surfactants that are mainly chemically derived and petroleum-based. Next, we see oleochemical surfactants that can be biodegradable but still have petroleum content. And then the third category, or the small sliver that you see, are called biosurfactants. And this represents biosurfactants that are either chemically or biologically derived. However, within this, there is still a differentiation. What we see is that the chemically derived biosurfactants still contain palm derivatives. And as we now transition to a more renewable economy, we see that a lot of end user formulators transitioning away from palm derivatives and wanting more natural alternatives, which is where we are in microbial biosurfactants. So within the surfactant industry, we have four different types, mainly non-ionic, cationic, anionic, and amphoteric. The most amount of surfactants are within the non-ionic sector, and each of these surfactants represent a different type of molecule that is best suited for specific end user formulations. Some might be better suited for detergents, while others might be better suited for shampoos or conditioners, for example. Now, currently the problem is really that synthetic and even the eco-synthetic biosurfactants are derived from non-renewable petroleum sources. On the other end, we have natural biosurfactants that are costly and lack a range of options. At Dispersa, our goal is to solve this by bridging the gap in the available technology, by offering a unique platform of novel molecules that are natural and cost-efficient biosurfactants. We do this through our proprietary process that we're developing called BioEterna, where we use novel microbes and then go through the process of fermentation extraction, where we cultivate and collect these biosurfactant ingredients. These are soapy compounds that are functionally the same as chemical surfactants. They are non-toxic, palm-free, and biodegradable. How Dispersa differentiates our biosurfactants from what's on the market currently are really based on these three main pillars of compatibility by offering a range of biosurfactants. We offer novel compounds, so it's an IP advantage and new market opportunity, as well as high performance ingredients. And now, as we look forward to where we will be entering into the market with our ingredients, with the rise of the pandemic, we saw that there was an increased demand for cleaning products, sanitation products. We also saw that there was an amplified interest towards transitioning towards natural ingredients. And so it made the most sense for us to enter into this market initially of cleaning and personal care. With this, we launched PuraSurf, our first line of formulations and ingredients containing biosurfactants intended for this market. Here we work with end user manufacturers like cleaning product companies, for example, who wish to up their sustainability factor by including and replacing their synthetic surfactants with our natural alternatives. To date, it's been over 1.5 years since inception of Dispersa, and we couldn't have done this without the generous support of our partners and supporters. We have secured 2 paid pilot projects with first customers in the consumer packaged goods area, and later in the year, we will be scaling up towards our 400-liter capacity. We have also secured over $1.5 million in government funding and in-kind contributions. Next, we have our team. Together, we have experience ranging from microbiology, chemical engineering, chemistry, and business development in the clean tech sector. We're also currently hiring, so do check us out or feel free to send me an email if you're interested in joining our team. Together, our vision is to be a global leader in the surfactant market by advancing natural solutions one microbe at a time. Thank you so much for having me today.
Thank you so much, Nivatha, Adam, and Jennifer. Let's go right away with the questions, and we'll start with you, Adam. Tell us about the production of PHA plastic. When this is produced at Genecis, do you use 100% of the food waste? And if not, what happens with the rest of the food waste that you take as an input?
No, I mean, typically the conversion efficiency going from the solid, well, rather wet solid food waste to PHPV is somewhere between 2 and 4%, which is actually quite good compared to the conversion efficiency you would get even from something like corn or sugar. So that's quite good considering how rich those carbon sources are. But the food waste actually, so the food waste is fermented, produces VFAs, and then the leftover broth that comes out of these fermentation tanks will be used by anaerobic digestion plants. This is our current business model, so that they can produce methane from it, because it can still be used to produce methane.
Cool, amazing. Thanks for sharing that, Adam.
Sure.
I have another question for Nivatha. So, and I actually found this question in the FAQ from Dispersa.ca, but I really like it. So, are they microbes or bacteria in your final product? Can you tell us more in detail about this process of getting from the microbes to the final soap product, biosurfactant?
Yeah, absolutely. So our final product in the end contains no microbes. In fact, similar to Genecis, we essentially have our microbial fermentation process, and then we go through extracting the actual byproduct that is a result of what the microbes actually produce, which is a soapy compound called biosurfactants. In the end, the ingredient that we have can be in either a powder or a liquid type honey form, and this is what in turn that the end users then integrate into their own formulations, for example. They contain no microbes.
All right. Thanks, Nivatha. And another question now for Jennifer. So from what I understood from your presentation, the milk at Real Milk is real milk. It's not a substitute of milk like coconut milk or almond milk. Can you tell us more about what you expect to be how do you expect to be the final product from your milk? Is it molecularly different than one that we would obtain, let's say, if we collect it fresh from a cow?
Actually, Bettermilk's milk will behave, taste, and feel exactly like traditional milk, which means that once inserted into dairy products, will have the same texture, the same feel, and the same chemical functionality as real milk. So in that case, I think the best question to ask is not if it's molecularly different, but if it's functionally similar to real milk, and it's exactly the same. So yeah.
Okay, thanks for sharing. I'll go back to that in a second. To your point, very interesting. But I have a question for Adam from the audience, and is from Varsha. Adam, Varsha would like to know if your company has a scope for partnership of funding with PhDs working in other industrially biopolymers.
I mean, currently our primary focus is on PHAs, so I don't think we're going to be looking at producing anything else in the next few years. And it would, of course, have to, we'd have to do some kind of economic market analysis on this to see what the market is, how profitable it is. I have to actually look at how we can produce this polymer in an organism and whether it's compatible to the carbon sources we want to use. So the short answer is it's complicated. So, I mean, it's possible, but right now we're really not. I mean, I used to make hyaluronic acid when I was a PhD student, and that's something we've thought about, but we're really focused on what we're doing right now. So in the short term, I would say that's not really something we're looking into.
All right. Thanks, Adam. And Jennifer, I'll take it back to you to the point where you were saying if it's functionally similar. I wonder, and we had this discussion in the panel before, but I'd like to have your thoughts, and these are on consumer acceptability. Can you tell us about that in your case for Bettermilk? Have you done any studies, or do you have any expectations on that?
Yes, that's a really good question. Right now, we have a team in Bettermilk that are focusing on consumer acceptability of cell-based milk. We're basing on research on previous research that has been done with cell-based meat, in which consumers feel that they will likely consume cell-based meat if they are educated about the process, if they're certain about the safety of the food products, and also if the price is low and if the market share is big, which means that as more people will start to consume the products that are cell-based, more consumers will be likely to accept it.
Okay, yeah, and I imagine this will happen probably at the same time with many different products. So the education in the public will happen at the same time with milk,
Yeah.
with meat, with other types. So that's good to hear you getting that positive feedback as well. I'm not going to ask this question to Nivatha or Adam unless you can tell me that you have had some negative impact in the production of the surfactant or of the plastics. But my assumption is that in your case, you're actually using the way you're producing this as a leverage for your consumers. Can you comment on that? Perhaps first you, Nivatha?
Yeah, absolutely. So is this in regards to like the actual process or the product, I guess? Yeah, absolutely. So, I mean, what we're producing are palm-free natural alternatives to synthetic surfactants. As we scale up, of course, with the bioprocesses, I think for all of us, there's always the unique challenges that we have as we scale up step by step. And for us, as well as we're now transitioning towards a lab scale and going towards our demo scale, we're really looking forward to that. It's definitely going to be an adventure, but a challenge accepted.
Cool. Thanks for sharing. And a follow-up on that, at the moment, are you planning to expand the use of surfactants to remediation or cosmetics, or for the moment you're focused more on the soap part?
Yeah, absolutely. So for the moment, we're focused on the consumer packaged goods area, just because we've seen that there is a rise in interest for natural ingredients. But in the future, we will be diversifying into other markets like oil and gas, agriculture, and more.
Okay, thanks. Looking forward to that. I have here a question from Julia to Nivatha and Jennifer. And Julia is wondering, what was your experience going from conception to medium scale-up work? Maybe you can start, Jennifer.
That's a good question. We aren't ready right now to have huge quantities of medium since we have little quantities of cells. Maybe Nivatha is closer to this final project as I am, so I will leave the floor to Nivatha actually.
Yeah, thanks, Jennifer, and thanks, Ana. We're currently producing our molecules at the lab scale, and now we're going to be going into using bioreactors. And so although we collect these materials at the end, of course, as we scale up, as I mentioned, there will be numerous challenges and optimization that's required at every step. Similar to what Jennifer has said.
Thanks. I have another question for you, Jennifer. It seems that people are interested in learning about the Bettermilk. So there's a question from Andy, and he's wondering if the milk from Bettermilk is going to be capable of producing other products like cheese, butter, and I don't know, ice cream, etc.?
Yes, absolutely. So what we're aiming to do is sell our milk to food companies who use dairy ingredients in their food. Like you mentioned, for ice cream, for cheese, we can even sell it to protein companies who use it to make protein shakes. We can even go as far as putting it in chips or any kind of foods that uses dairy products inside of their products.
Cool, thanks. And there was a follow-up question on that from Christos. He wanted to know if the milk from Bettermilk is suitable for people with, I guess, lactose allergies, so dairy allergy.
Yes, absolutely. Like I mentioned in my presentation, our milk is customizable, which means that we can tell the cells to produce no lactose in the milk. So yes, it will be lactose-free if the consumer wants it to be lactose-free.
All right. Thanks for sharing. I'm thinking back to Adam. And tell us a bit about your work at Genecis. I saw from your title that you were an engineer at Genecis, and I would like to know what type of organisms do you engineer or have you engineered at Genecis?
Well, they're bacteria. So, before I came to Genesis, I was working with Bacillus and E. coli, and now we're mostly focused on E. coli because, it's got a proven track record for producing PHBVs, and Bacillus, we had hopes, but it doesn't seem to do it very well. So we're going with E. coli. But obviously, our focus is on microbes because producing plastics and higher organisms just isn't. It's not feasible.
Right. Thanks. And a follow-up on that, because there's a lot of buzzwords in the clean tech sector. And especially you were mentioning about bioplastics and the difference between pH plastics and bioplastics versus biodegradable plastics. Can you tell us just a very short definition of this difference between all these plastics for the consumer? What should I be looking for when I get a bioplastic that is biodegradable, for example?
Well, I mean, a bioplastic would have to be produced by a microbe, and therefore it would be biodegradable. So PHAs are the original bioplastic. Obviously, if you're looking at something that's produced sustainably, then preferably something that's produced in a cell. But anything that's biodegradable is better than a petroleum-based plastic. But certainly, if you can produce it in a cell, then it's likely going to be a more sustainable process than if you're doing it synthetically, chemicals. But anything that's biodegradable is still much better than polypropylene or polyethylene, that kind of thing.
Okay, so they're synonyms. So a bioplastic is always biodegradable?
Yeah, and it would be produced inside an organism. It's produced inside a cell. So certainly, this isn't the only example, but again, PHAs are probably the best known example and the most versatile in terms of their properties and how we can tailor the properties because, PHBV is just one copolymer. There are a lot of other monomers that you could put in various combinations to produce plastics with different properties. So, they're versatile and they're, the pathways are pretty well understood. So, yeah.
All right.
Definitely a
Thank
bioplastic,
you.
though. Yeah.
Thanks, Adam, for clarifying that.
Sure.
I found that was very confusing. Just to go to a more general question, and since some of the topics of the conference that we're having, it's on talent, on specialized talent, on biology and bioinnovation, I wanted to ask this question to Nivatha and Jennifer. On your experience about growing your team and finding a specialized talent that knows of bioreactors and fermentation and so on, or even mammary cells to produce milk. How has it been easier for you to grow your team and find people to join your team? We can start maybe Nivatha. If that's
Sure. Yeah, absolutely. I think that's a great question. And being within the biotech sector, but specifically in biomanufacturing, I think hiring tech talent is definitely the main component in advancing any of our product development. Our first hire was tech, and even now we're expanding our tech team. And I think especially on our end, we're coming from different angles. We like to have a team that's very diverse in terms of background, coming from different majors, for example, not only chemical engineering or having fermentation experience, but also synthetic biology, molecular biology. And so we're definitely trying to create that sort of well-rounded team as we continue to develop and go on to different stages in our product development. At the moment, given that we are going to be scaling up our process, we are looking for example, interns who have interest in molecular biology, as well as down the line we'll be hiring more for fermentation roles as we scale up and have that technical need for process engineers, for example.
All right. Thanks for sharing. Jennifer, would you like to comment on that too?
Yes, very similar to what Nivatha was saying. More hiring is so important because it makes your company. What we've noticed is that exactly like Nivatha says, we're not exactly looking at someone who made milk from mammal cells because that doesn't exist. So what we've been doing is just looking at the interest of the actual person and looking at innovative ideas and just how they apply their research to our company. And I love this part. It's just amazing to meet so much people who want to change the world the way we want to do it.
Thanks for sharing. We have a video question that we wanted somebody submitted to us for the three of you. Can we have that asked?
Hi, I'm Farah Benamain from Montreal in Vivo, and we're the Life Sciences and Health Technologies cluster of the Greater Montreal area. We're an economic development agency whose mission is to foster the growth and competitiveness of companies in our sector. And typically, we've worked most often with companies that are active in human health, which doesn't mean we're not interested in engaging more closely with companies in the life sciences sector more broadly. So the question that I have for you is, how can we do that, and how can we better support organizations like yourselves? Thank you.
Thank you so much, Farah, for submitting that question. Who would like to start? We have a volunteer. Otherwise, Adam, we can start with you.
Well, I mean, I'm not on the financial end of our business, but I mean, unfortunately, in general in Canada, there's not the investment in biotech beyond the medical field that there really should be, and you know, a lot of that just has to do with you know, I guess education of the larger population and even people in the government. I mean, I don't think many politicians really understand or know what we're up to, and so they need to understand how important the work that we're doing is. And unfortunately, there seems to be this kind of tug of war between accepting that, you know, the planet is in trouble and not, or just saying, well, you know, we have an economy to maintain and people have to survive, and so that's the priority. So I think accepting that we're heading down a road that's one way, and then understanding that the only way to resolve you know, these problems with global warming and plastics taking up our oceans is that we have to take action, which requires funding. And you know, it can start with the government, it can move on to private enterprise, because once you get the private sector on board, then the problem is solved, right? You get basically unlimited funding. So, but it starts, I guess, with government and awareness. It's my interpretation. So.
Yeah, thanks for sharing that. We actually have a panel right after this on that topic. Thanks for sharing, Adam. Nivatha, Jennifer, do you want to share your thoughts on that question?
Yeah, I agree completely with Adam. I think, yeah, it was on point.
Nivatha, something to add?
Yes, really. I guess just to add, I think that especially specific to Quebec, there are more and more programs, and I think it's definitely headed in that direction towards helping to stir up and support the bioeconomy. I know that we have a few organizations, for example, the recent announcement on Fonds d'Action to help support the circular economy and develop startups in this sector. So definitely excited to see the momentum keep up and to continue.
Great, thank you so much, the two of you, for presenting your startups and for sharing your thoughts. I'm gonna wrap it up here.